Vectors for cloning genes in plants and animals. So what do you mean by vectors for cloning genes in plants and animals? If you take the plants normally, this Agrobacterium tumefaciens, right? So this Agrobacterium tumefaciens, it causes a disease that is called the ground high disease in most plants. So actually it causes a tumor like. So naturally it is causing the tumor. And even if you take the retroviruses, so what is the role of retroviruses? They are also carcinogenic or cancer causing in animals. So these Agrobacterium tumefaciens and retroviruses, naturally they are able to induce the tumor, right? So they are actually the pathogenic. So now what the scientists are doing is to remove this virulence or the most disease causing part of it, because they are able to introduce the genes actually, right? Some other virus is able to introduce the genes into the animals. So what they are doing is to remove this virulent or the disease causing genes, and they can be converted into a useful vector, because they are able to introduce this genes into a different organism or a host organism, right? So the genetic tools of some pathogens. So these pathogens are having the genetic tools, and it is transformed into useful vectors. Actually, it is pathogenic and you're converting it into a useful vector, right? Because these pathogens, they are able to introduce this carcinogenic genes into a animal or the tumor causing gene into a plant, and they are transforming this into a useful vector for delivering genes to plants and animals. Okay, so here this is called disarmed, that is we are destroying the disease causing effect of that particular virus or the particular organism. Okay, so here the Agrobacterium tumefaciens, it can deliver a piece of DNA to normal plant cells, right, into a tumor. So this Agrobacterium tumefaciens, actually they have the capability of converting or transforming the normal plant cell into a tumor cells. And these tumor cells, they produce the chemicals required by the pathogens, right? So now this tumor-inducing T8 plasmid of Agrobacterium tumefaciens is modified into a cloning vector. We are transforming it into a cloning vector, which is not pathogenic. But what are the aspects that we are using? That we can use the mechanisms to deliver the genes of interest into the plants by using this mechanism, right? Similarly, in the case of retroviruses in animals, they can transform the normal cells into cancer cells. So they are used to deliver the desirable genes into animal cells. So Agrobacterium tumefaciens, it is used to deliver the genes of interest into plants, and retroviruses are used to deliver the desirable genes into animal cells.